In this video, we explore how we go from an empty plasmid vector to a so-called construct that is a plasmid that carries a gene of interest. We're going to walk through some of the fundamental steps for this to provide an overview. In order to do that, I'm going to go ahead and bring up the whiteboard so that we can sketch some things out here. So we're going to try to, by the end of this video, answer the question of how are plasmid constructs created. In the last video, what I did was gave you this overview schematic of taking an empty plasmid, shown here on the left, and we introduced a gene of interest into that plasmid to create the so-called construct. We had our empty plasmid on the left, the construct that we've constructed with the gene of interest on the right, that gene of interest being from some other organism that is perhaps hard to work with in the lab. And so we introduce that gene of interest into this plasmid so that ultimately we can introduce that construct into some host such as E. coli and produce lots and lots of the protein that corresponds to that gene. So how do we go about going from the empty plasmid starting material to our construct? That is what we are going to learn here. So our empty plasmid, which we call PXYZ here, typically those are purchased from a commercial vendor. There's a variety of vendors that sell plasmids that are these circular DNAs that contain an antibiotic resistance gene. Additionally, what those plasmids also typically contain is what's referred to as a multiple cloning site, or an MCS for short. So I'm going to draw a little abbreviation there of a box and label that as the MCS, the multiple cloning site. The multiple cloning site is the location of the plasmid where the plasmid is ready to accommodate the foreign gene. And at the multiple cloning site, the way that it is able to accommodate that foreign gene is that the multiple cloning site generally contains multiple restriction sites, multiple DNA sequences at that location that would be recognized by a so-called restriction enzyme, which you can think of as being like DNA scissors. Restriction enzymes cut DNA at very specific nucleotide sequences. Restriction enzymes are commercially available. And so what would happen here is that the multiple cloning site has multiple well-known restriction sites where a restriction site is defined as a specific DNA sequence, usually of about 4 to 6 nucleotides, which I'm going to abbreviate as NT, that is recognized by one or more restriction enzymes. And getting into some more jargon here, the restriction enzyme is an enzyme, a catalyst, that specifically recognizes individual DNA sequences and cleaves the DNA at that particular location. So a restriction enzyme is going to recognize and catalyze cutting or cleavage of DNA at the specific sequence that it recognizes. And usually that specific sequence that it recognizes is that 4 to 6 nucleotides in length. And so the multiple cloning site will be a short region within the plasmid that has multiple locations that are recognized by different restriction enzymes to give the experimenter some flexibility about what restriction enzymes they want to use to cut that DNA. Additionally, very often, plasmids will also contain, immediately upstream of this multiple cloning site where we're going to be cutting the plasmid open and introducing the gene of interest, there will also be such things as promoters and ribosomal binding sites, which are other essential elements that enable that gene that we'll be inserting to be adequately transcribed and hence translated. So the multiple cloning site is going to be ultimately the location where the gene of interest is introduced into the plasmid. And the reason why that multiple cloning site generally has a bunch of different restriction sites is because the way that we are going to introduce that foreign DNA is we will initially use restriction enzymes to cut open this circular empty plasmid. We will then stitch in, or ligate as the technical term is, the gene of interest into that opened up linearized plasmid. And then we'll reseal the resulting DNA sequence back into the cyclic form that now has the gene of interest in it. So how do we go about doing that? That is what we are going to look at next here as we continue onward with this. So continuing onward with this, I'm going to just zoom out a little bit onto what I've already written there. So we still have it for reference, but it's not front and center. So in order to introduce that foreign DNA into the empty plasmid, into our vector, what we will do first is cut open that vector, that empty plasmid, using restriction enzymes. So cut open the plasmid that is empty with restriction enzymes. And those restriction enzymes are ones that will cleave DNA at the multiple cloning site. So those restriction enzymes, if we go back to our discussion of nucleic acids, what the restriction enzymes are doing is they are breaking phosphodiester bonds to cleave the DNA of the strand. They are going to break the DNA of the strand so that you no longer have a circular DNA, but instead it's linearized. And so doing that schematically, what we would be looking at, we have our antibiotic resistance gene that's there, our multiple cloning site in red, and when we treat the plasmid with one or more restriction enzymes that recognize DNA sequences in that so-called multiple cloning site, what's going to happen is that the restriction enzyme is going to cut in that multiple cloning site. So the restriction enzyme will cut in that multiple cloning site, and that's going to result in the DNA being linearized. Now, once the DNA is linearized, based on the way that most restriction enzymes work, not all of them, but most, cut the DNA in a way that leaves so-called sticky ends. It leaves an overhang at one end of the DNA. So we keep in mind that DNA is a double helix. So when we make that cut with a restriction enzyme, we're not just in grand total cutting one strand of DNA. We have two strands. These would be the two hydrogen bonded strands. So this would be like your double helix coming through here, all those hydrogen bonds all along that I'm drawing in a very simplistic way. And then once the restriction enzyme has come in and cut the initial circular DNA, that linearizes it, but it leaves the double helix there in the middle. At the ends though, there will be so-called sticky ends. These are slight overhangs at each end, at this end, and at this end, at the 5' end and the 3' end, where there are a few, generally 4 to 6 or 7, nucleotides hanging off the end here that are not hydrogen bonded to the other strand. They're just dangling there, and they're called sticky ends because they are eager to hydrogen bond to some other complementary sequence. And so, for example, just to fill in some possibility here, we could say that the sticky end at this end is TTAA as the sequence. And the sticky end at the other end is AATT. And yes, those are complementary at the two ends, here and here, because they originated by breaking the DNA at the position here and here. So if you imagine that this was going to recyclize, the 4 nucleotides here are the ones that are complementary to the ones at the other end. Those are the ones that would have been hydrogen bonding to create the circular DNA that was the original plasmid. So now that we have this linearized plasmid with sticky ends, those sticky ends are eager to find mates. They are eager to find DNA that is complementary to that. And so what we can do is create a gene insert that has sticky ends that are complementary to the ones at each end of our cut plasmid. So step 2 in the process, which in actuality can be done simultaneous to step 1 if we're working in the lab, these can be carried out at the same time, is that we need to create the gene of interest with sticky ends that are complementary to our cut open plasmid or our cut open vector. And there are 3 ways that we can go about creating that gene of interest. Actually, there's probably more than 3 ways, but there's 3 primary ways. One of those ways is that we can buy so-called synthetic DNA. Synthetic DNA is DNA that has a specific sequence that we can order from a scientific supplier. So by ordering it from a scientific supplier, we can obtain DNA that is of any particular sequence that we want. And so if we have the full genome sequence of our organism of interest, we can order a DNA sequence that matches the gene from that organism that we're interested in, obtain that, and we can have added to the ends of that gene sequences that are complementary to our sticky ends here. So that they will be very eager to come in and link through hydrogen bonds there and here. So you can buy synthetic DNA of the gene of interest. So that would be our green DNA here, which will have sticky ends that are complementary to those of the plasmid that we've created. Another way to go about doing this is, rather than buying synthetic DNA, you can also amplify by using the polymerase chain reaction, PCR. You can amplify the DNA of interest from the original organism, from the organism that is the original carrier of that gene. So I'm not going to go into PCR, but think of it as a DNA copy machine where you can take from a few nanograms of DNA, you can amplify a specific targeted sequence, such as the sequence of a gene, by many millions fold to create a large amount of that particular DNA sequence of interest for cloning into the plasmid vector. And that PCR strategy will also allow you, through the PCR experiment, through the amplification of DNA, to append on that gene of interest whatever sticky end sequence you want. So you can bring in ends that are complementary to our plasmid here. So you can amplify the DNA from the organism of interest. The other strategy, and these days probably the least used strategy of these 3, because PCR is now cheap and generally effective, synthetic DNA is becoming increasingly cost effective as the years go on. But an old school way of doing this, in a way that's still done to some extent, is that if you're lucky and the gene of interest has restriction sites that are recognized by the same restriction enzymes that would recognize and cut to make our vector sticky ends, you can restriction digest the gene of interest out of the original organism so that you will have that fragment of DNA that has the particular sticky ends you want straight away. But it's kind of a unicorn to be able to find a gene of interest that immediately at the 5' end and 3' end, or anywhere close to the 5' end and 3' end of that gene, has restriction sites for the same restriction enzymes that are found in suitable for the multiple cloning site. So one way or the other, you need to create your gene of interest. That gene of interest needs to have sticky ends that are complementary to your cut vector. Then from there, what you do is make a match made in heaven. And that is, you need for the cut plasmid to form a union with the gene of interest. And so the way that we go about doing that is by using an enzyme referred to as a ligase. A ligase is going to be the catalyst of the phosphodiester bond formation that is needed in order to link covalently the DNA from our plasmid, which I'll go ahead and call this our cut plasmid DNA right here. We need to be able to link that to our DNA from the gene that has complementary sticky ends here and here that are complementary to the sticky ends here and here. And the enzyme that we use to catalyze that reaction, because those phosphodiester bonds are not going to form spontaneously, is a ligase. So ligase is going to catalyze the formation of our phosphodiester bonds. Stitching in covalently the gene into the plasmid and reforming circular DNA to give us the construct. So end game, you will have your part in blue that originated from the empty plasmid, or the vector as we could call it. Then we will have our gene in green that we have inserted that to give us, as our grand finale here, the construct. And keep in mind that construct would have the elements that we originally had in the plasmid, such as if our plasmid had an antibiotic resistance gene, that's going to get carried through into the final product here as well. And keep in mind that although in the abbreviated form of plasmids, we generally just show a single line going around that circular DNA, this is actually double helical DNA all the way around. So keep in mind this would be a double helix. The same was true throughout. So at the very beginning here, this empty plasmid was a double helix as well. It's just for simplicity purposes. When people make plasmid diagrams, they usually don't show it as two strands of DNA. They just show it as a simplified representation of one line or one circle there going around. So in the next video, what we are going to focus on is, now that we've created our construct, how do we go about introducing that construct into a host cell so that we can actually multiply this construct, and so that we can use the machinery of the cell, namely the ribosome, in order to take the gene sequence and ultimately translate that into a protein sequence so that we can work with the protein. So that is what we'll get into in the next video. Stay tuned for that.